All right. Hello, nutrition students. So this week we'll be moving on to our second chapter, looking at our different macronutrients. This time it'll be on lipids. And so in this first part, we're going to look at just the different types of fatty acids and talk about which lipids are dietary versus non-dietary. So all the fats that we're getting in our diet, the fat that you see described on your nutrition facts panels, that is referring to lipids. And so these are large macromolecules that do not dissolve in water. And we have three types of lipids that we can find in our food. The most important to get from our food is going to be triglycerides. These are our dietary fat. So anytime we're talking about a dietary version of a nutrient, that is the type that we must get from our food. Our body cannot synthesize it or create it. Our cells cannot make them on their own. And so we get those from our diet, from what we're eating. And so the only dietary fat, the only version of a lipid that we are getting from our food that we cannot synthesize ourselves are triglycerides. But we're also getting phospholipids and sterols, but these are non-dietary fats, again, meaning that our cells can create their own phospholipids and create their own sterols, but they cannot create triglycerides. So in order to get triglycerides, we must consume them. And all lipids are composed of the monomers a glycerol head and fatty acid tail. Triglycerides specifically have three fatty acid tails, hence the prefix tri. Okay? So triglyceride has a glycerol head with three fatty acid tails. And then it is the structure of these fatty acid tails that is going to differ between the different types of lipids or the different types of fats, and thus is going to change how our bodies can use them, and thus the health effects that those fats might have. So fatty acids can differ in the length of their carbon chain. So we can have short, medium, or long chain fatty acids. The degree of saturation of that fatty acid tail, and we'll look at a few slides referring to saturation, but this is talking about how many hydrogen atoms surround each carbon. And so again, we'll come back to this, but all fatty acid tails, right, are just long chains of carbons with hydrogens branching off of them. And so the number of hydrogens that are bonded to that carbon is what is going to change the saturation. So again, fatty acids can differ in length, degree of saturation, and then lastly, their shape. And that shape is going to be determined by how saturated those carbons are. So the major difference, or the major categories that we'll talk about relating to fatty acids, is saturated versus unsaturated. So a saturated fatty acid tail has all of their carbons fully saturated by hydrogens, meaning that each carbon is only giving up some of its bonds to the other two carbons that are involved in that chain. And then the rest of their bonds are being taken up by hydrogen. So each of the carbons along this fatty acid chain have one bond with the previous carbon, one bond with the next carbon, and then two bonds being given to individual hydrogens. So each carbon here has two hydrogens attached to it, except for the very last one, which has three. An unsaturated fatty acid has at least one double bond between carbons along its chain. And since there are two bonds being held between these carbons, there's only one available bond for hydrogen on each of the carbons involved. And so I'll show another diagram that I think shows this a bit better. But again, the difference between saturated and unsaturated is that a saturated fatty acid is fully saturated by hydrogen. An unsaturated fatty acid has at least one double bond between their carbons, and thus will have less hydrogens. And so beyond that, we have mono unsaturated fats or fatty acids, meaning there is one double bond in their chain. And then we have poly unsaturated fatty acids, which have two or more double bonds along their carbon chain. So again, saturated fatty acids have hydrogen atoms surrounding every carbon in the chain. Mono unsaturated fatty acids lack the hydrogen atoms that would have been there in one region because there is at least one double bond between carbons. And then poly unsaturated fatty acids lack multiple hydrogen atoms in multiple regions wherever there is another double bond. So I think this diagram shows it better, right? So carbons have 4 valence electrons, and thus they have 4 available bonds that can be made with other atoms. And so in a saturated fatty acid, each carbon is giving 1 bond to the carbon before it and 1 bond to the carbon after it. And so its 2 remaining available bonds are being taken up by hydrogen. This creates a very stable, almost linear structure to the molecule. Unsaturated fatty acids have a double bond, right? So 2 of the bonds from each of these carbons here are being given to each other. So there's only 1 available bond left for a hydrogen. And so in this case, a mono unsaturated fatty acid like this one, which only has a single double bond in it, this molecule would have 2 less hydrogens than the equivalent saturated fatty acid. Because 1 bond from each of these carbons would have been given to hydrogen, but since they are double bonded instead, there are 2 missing hydrogens from this fatty acid tail. Okay. And so that saturation is going to determine the shape of these fatty acid tails and have a large impact on how our body can use them or how they interact with the environment. So saturated fatty acids can be really tightly packed together. Right? So if we look back at this saturated fatty acid is almost completely linear in shape. And so if you imagine multiple of these stacked on top of each other or next to each other, that creates a really solid, stable structure. And so these saturated fatty acids tend to remain stable and remain solid at room temperature. And we get a lot of saturated fatty acids from animals, right? The only exception really being fish, which produce a lot of oils that are mainly unsaturated fats. But most of the animal fat that we are consuming is going to be saturated. So think of all of your dairy products, right? So butter, lard, right? These are saturated fatty acids. Unsaturated fatty acids, because of that double bond, creates a kink in that tail, so a slightly different shape in the tail, and thus do not stack. They do not remain in that solid structure. They have some bends and bonds. So if you imagine like building blocks, if you're trying to stack them on top of each other, flat blocks are going to be much easier to stack compared to bended or anything with a kink or turn in it. It'd be harder to stack. And thus, unsaturated fatty acids have more room in between individual fats and thus are liquid at room temperature. So think about your like oils and vegetable oils that you use to cook with, right? You can keep them out on your counter and they will remain liquid. And so plant oils are a great example of foods really high in polyunsaturated fatty acids. Okay. And so now we can go a little bit further. So within these unsaturated fatty acids, not only do we have monounsaturated and polyunsaturated fats, but each of those double bonds, that is connecting those 2 carbons, they can either be cis or trans bonds, and thus introducing trans fats. Right? So most of the unsaturated fats that you see described or measured in your food, that is referring to the cis trans fats, where the hydrogens in that carbon chain are on the same side, and thus giving that kink and creating that liquid state at room temperature. But trans fats also still have that double bond. However, the hydrogens that are bonded to those carbons are on opposite sides. And what this does is it essentially nullifies the kink that would have been there in a typical unsaturated fat, because the hydrogens are on opposite sides. That causes the chain to go back towards an almost linear structure and thus become more solid. And this is a purely artificial product. There are no natural sources of trans fats. This is a result of creating hydrogenated fat, or essentially adding hydrogens to plant oils, and thus producing our like fake butters or margarine. Right? So those were originally plant oils. They're not animal fats. They were plant oils that we have turned into a solid or semi-solid by adding hydrogens and thus creating these trans fats. Okay. So that's a process called hydrogenation, the addition of hydrogen atoms to unsaturated fatty acids. And this converts these liquid vegetable fats and oils into a more solid form, thus increasing their shelf life, preventing rot, and improving some of their baking properties. Okay. However, and so trans fats are really the only type of fat that we have solid scientific research to indicate that they are unhealthy. So out of all the different fatty acid tails that we've talked about, all the different fat types that we just introduced and you've heard of, or we've talked about in this class before, all of those are generally healthy, or any of the research that said they were unhealthy has been shown to be inconsistent or untrue. The only type of fat that we have solid evidence to say is unhealthy and you should try to avoid is for trans fats. And we made them, right? And that mainly comes from the fact that they are likely to impact our production of these different lipid products. So after we digest it and absorb these fats into our cells, we then change them into different lipoproteins, different types of lipoproteins. And those different types of lipoproteins are either more likely to deposit cholesterol onto our cell walls or less likely to deposit cholesterol, and thus will make it to their destination. And trans fats are much more likely to convert our fats or be absorbed as low density lipoproteins and decrease the amount of high density lipoproteins that we have in our blood. And so in general, high density lipoproteins are healthier and safer for us. Low density lipoproteins are the version of lipoprotein that is likely to deposit cholesterol into the linings of our blood vessels, our arteries, and thus create those clots, those plaques. Here's a depiction of what I was talking about with this cis versus trans arrangement. So natural unsaturated fats are in this cis formation. So we have our double bonded carbons, and then each carbon has one hydrogen attached to it. And those hydrogens are on the same side of this structure. And thus that pushes the shape of the tail to create that kink that I was talking about, that bend. Right? Trans fatty acids, we still have a double bond. However, the hydrogens bonded to these two carbons are on opposite sides. And thus that essentially reverses that bend, this kink, and puts it back into that almost solid, rigid structure, right? That is much more structurally stable. And so it can turn these liquid oils back into an almost solid fat. Right. So here we have a look at different food products and the concentration of these various different types of fats that we've been talking about. And so we also get a short introduction here to our omega-3 and omega-6 fats. We'll talk more about those later, but these are examples of common polyunsaturated fats that are generally considered the healthiest or the most likely to be converted into high density lipoproteins, and thus the least likely to deposit cholesterol into our arterial walls. So animal fats and tropical oils such as coconut and palm oil are mostly saturated fatty acids. Okay, so we see that depicted in red, with the blue being our monounsaturated fats, and then green being omega-6 and yellow being omega-3. So coconut oil is actually really, really high in saturated fat with a little bit of our monounsaturated and a little bit of omega-6. And then we have butter, beef tallow, right? Some animal products, lard, animal products, also very high in saturated fat, with some monounsaturated fats, but not very many polyunsaturated fats. So these are all much more likely to be solid at room temperature. And then some vegetable oils, specifically like olive, canola, and peanut oil, are really rich or have a high concentration of monounsaturated fats. So where there is a single double bond along their fatty acid chain. But most vegetable oils are really high in polyunsaturated fats. So these omega-3s and omega-6 fats here. You can see those much larger portions being taken up by the polyunsaturated fats and very low proportions being taken up by saturated fats. Right. So a few practice questions here to kind of review what we've talked about so far. So take a second to read each of these and think about your answer and then continue. So in this case, the best way to describe a fatty acid that has one region of unsaturation is, hopefully you chose mono unsaturated here, right? Mono is a prefix meaning one. So a mono unsaturated fat, has one region of unsaturation. Poly is a prefix meaning many, so a polyunsaturated fat would have many regions of unsaturation. Right. When a fatty acid is described as being saturated, it means that, pause, think about your answer. So hopefully here you're able to identify that A is the most likely answer. So a saturated fat contains only single bonds between its carbon atoms. Right? Or you could describe a saturated fat by saying that all of the carbons in the fatty acid chain or in the fatty acid tail are completely saturated by hydrogen. Right? So there are only single bonds between each of the carbons in a saturated fatty acid. Okay. A fatty acid that has 2 double bonds would lack how many hydrogen units? Just take a second to think about how many hydrogens get lost for each double bond that shows up in the chain. And hopefully here you're able to identify that 4 would be the answer. So let's go back a bit. I guess this picture works fine. Right? Or no, let's go back to the slide that I think does this best. Right? Okay, so here when you can compare these carbons in the center here, or look at this bond, right? When a double bond forms, a hydrogen is being lost from both of the carbons involved in that double bond. So this carbon would have typically had a hydrogen here. This carbon would have typically had a hydrogen here. And so one double bond removes 2 hydrogens. And so if you're looking at a fatty acid that has 2 double bonds, that means that they would have lost 4 hydrogens compared to a saturated fat. Right. Let's catch up where we were. All right. Just a couple more slides here. Now looking at phospholipids, and then lastly our sterols. So these are versions of lipids that our cells can synthesize. We can manufacture them ourselves, and so we don't necessarily need to get them from our diet, but we do get them from our diet, right? Because everything we're eating is made up of cells, and so those cells are going to have the same, similar, if not the same stuff that we do. And definitely phospholipids, because every single cell has membranes made up of phospholipids. And so a phospholipid is made up of the same basic stuff. It has a glycerol head, the same basic monomers. So a glycerol head with 2 fatty acid tails attached to it, and then a phosphate group. And phosphate is I guess technically a molecule. Sorry, I was trying to think if there's a more accurate word, but it is a small molecule that is utilized as for like transferring energy in most examples, but is also used in communication. Right? It just provides a slightly different chemical structure and chemical property to this lipid. But this is what makes up the membranes of our cells. Right? So we have all of these hydrophilic heads. The heads can interact with water. So these hydrophilic heads are pointing out towards the environment and pointing in towards the cytoplasm of the cell. And then these hydrophobic fatty acids, which cannot bond with water, they repel water, are all pointed inwards. And our cells can change the structure of these phospholipids in order to increase flexibility or fluidity of our membrane. So when molecules need to move along the membrane, such as moving cholesterol or moving some of the proteins that we have in our membrane, the membrane can, or our cells can make our phospholipids more unsaturated. So we can decrease the saturation in this region of the membrane, thus making this more fluid, more mobile, and so we can move these molecules across. This also plays a role in like protection and homeostasis, such as maintaining heat or reducing the amount of heat in that cell, etc. But the main point I wanted to show here is that our cells do have the ability to modify the fatty acid tails that are in our phospholipid bilayer. So we can change them from saturated to unsaturated. And then lastly, sterols. So again, another example of a lipid that our bodies can synthesize, so we don't necessarily need to get them from our diet. However, we do. These are lipids that contain multiple carbon rings, or essentially monosaccharides within their lipid structure. So this is what makes up cholesterol, which is a important component of our cell membranes. It also has a role or plays a role in the rigidity of our membrane. So the more cholesterol, the more structurally stable that version of the membrane would be. And then some of our hormones, such as testosterone and estrogen. And so again, hormones can be either protein or lipid-based. Testosterone and estrogen are examples of lipid-based hormones. Okay? So these are manufactured in our bodies, not essential components of our diet because we can make them ourselves. And that's where I'll stop for this video. I will see you guys in the next one.